Namaste everyone, welcome to Need Guidance. In this video, we are going to discuss about histology slides, how to identify the given slide, what are the basic features of each individual slide so that we can identify the slides fastly as the time is limited in the exam. Starting with tendon. Tendon is a special connective tissue. It is having mainly, we can find its structure is like this. Uh, there are specialized cells called as the fibrocytes. These are all the small fibrocytes. This is the main feature is that we can find this small fibrocytes. It's all about the tendon. I'm not taking much of the time in discussing as we have very short span of time in exam to identify the structures. Coming to the adipose tissue. As we have no adipose tissue, it can be identified easily. We have adipocytes in which the fats are stored. Excess fat in the body is stored in the adipocytes. It is all about adipose tissue. We have completed the one is the tendon and other one is the adipose tissue. Next, coming to hyaline cartilage. We are having three cartilage: hyaline, elastic, and fibro cartilage. Distinguishing feature of hyaline cartilage is that we can find staining actually in hyaline cartilage. Staining is well identified. We can see the purple color stain, and other thing is that see, we can see this purple color stain over here. And other thing is this ground substance is a homogeneous ground substance compared to elastic cartilage. And we can see the cell nest is clearly mentioned. Cell nest is having almost 2 to 3 cells. This is the features of hyaline cartilage. Main feature is that it is having homogeneous ground substance, and it is when the slide is focused, basically we have to see what is the ground substance, that is homogeneous ground substance, and the color of the slide is almost purple compared to other two slides, that is elastic and fibrocartilage, and the cells are arranged in cell nest. This is all about hyaline cartilage. Coming next to the elastic cartilage, we are having fibers are arranged in the hyaline cartilage. These are the small fibers, and the cells are arranged in one or two. Distinction feature is that in hyaline cartilage there was a stain, but and the ground system was a homogeneous ground substance because there is a same refractive index. And in elastic cartilage we usually find the fibers between the cells. Next, coming to the uh, fibrocartilage. Fibrocartilage, it is in fibrocartilage the cells are arranged in the rows. It's about fibrocartilage. We can, this is a slide of the fibrocartilage. We can find that cells are arranged in the rows between the fibers. These are all fibers. Cells we can find almost between feature is that fibers cells are placed within the fibers in almost they are placed in rows. This is fibrocartilage. We have completed three cartilages, two connective tissues. One is tendon, adipose tissue, hyaline cartilage, elastic cartilage, fibrocartilage. Uh, Next, we are discussing about TS of bone. TS of bone, we are having osteocytes. The circular shape is the eversion canal in which eversion vessels are found which supply the blood to the bone, and we are having a lamellae, that is the circumferential lamellae, interstitial lamellae. This is the TS of bone. Sorry, this is the concentric lamella. Sorry, this is the concentric lamella. We are having next between surrounding the eversion canal. We are having the concentric lamella. It's not circumferential; it is concentric. As it is concentric with the eversion canal, between the two between the two eversion system, we are having see what I am. This is the eversion canal. The one in yellow color is the the one in the red color is the concentric, that is next to the eversion canal, and the other one between the two osteons is the interstitial lamella. This is interstitial. circumferential is almost near the periosteum. It is about the TS of bone, which is the main feature of the TS of bone. Next, coming to the LS of bone. One more feature is that in TS of bone, Haversian canal is a main feature where we can find in the TS of bone. Workman canals are found in longitudinal section of bone. This is the longitudinal section of bone. We can find that these are arranged as we can find the workman's canal in LS of bone. Prominent feature of LS is workman's canal. Prominent feature of TS is Haversian canal. And cells are, these are the lamellae, which are, we can see them some straight lines. These are the lamellae. It's all about the TS of bone. Feature is that Haversian canals are arranged. Next, coming to the skeletal muscle. Skeletal muscles, muscle system we are having skeletal muscle, cardiac muscle, and smooth muscle. In skeletal muscle, we find mainly bundles of fibers. Surrounding each muscle fiber, we are having this is endomysium, and each of the bundle fibers are arranged by perimysium. This is perimysium, and wall of the muscle is surrounded by one sheath, that is epimysium. This is about the skeletal muscle. Next, we are having skeletal muscle, that is this is the TS of the skeletal muscle, and next is TS of the LS of the skeletal muscle. We are having longitudinal straight fibers, and main feature is that the nuclei in skeletal system, the nuclei is eccentric, means it is flat. It is flat, and we can find the striations. This is about the skeletal muscle LS. Next is cardiac muscle. Cardiac muscle feature is that we can find the branches between the fibers. These are all the, see, this is the branches between the fibers. This is the about cardiac muscle. We're having one mainly skeletal muscle and cardiac muscle. Next is about nervous system, peripheral nerve. We can find it easily in peripheral nerve, but some confusions are regarding skeletal muscle and peripheral nerve. In peripheral nerve, each neuron is having a covering that is called as endoneurium. This is endoneurium. Surrounding the each nerve fiber, we are having some bundles of fibers are surrounded by perineurium. This is the perineurium, and wall of the nerve is surrounded by epineurium. It's all about the peripheral nerve. Fibrocartilage, we have already discussed about it. The reason it is random means because in exam it is in a random way they have been put. Next is spinal ganglion. Spinal ganglion and sympathetic ganglion. In spinal ganglion, we have the nuclei, that is nuclei are found. These are pseudo-unipolar nuclei, in which nuclei is centrally placed and they are prominent. It's about spinal ganglia. Next is sympathetic ganglia. Sympathetic ganglia, the nuclei is almost eccentric, and fibers are scattered in the sympathetic ganglia. We can scattered fibers are there present in the, these are the scattered fibers, but in sympathetic ganglia, fibers are arranged in groups, and this is all about sympathetic ganglia. Next is mucous membrane, that is salivary glands, serous glands. Main feature is that in shortcut to say, these white patches, to remember I'm saying, these are more prominent in serous gland compared to others, because serous acini is more. This is about serous. Next is mucus. You can see main feature is that in a serous gland, these white spots are more. In mucus, the other type of acini, that is mucus acini, is prominent. It is the mucus acini. Next we are having mixed salivary gland. Here we can find both serous acini and mucus acini. These are serous, and we can also find the mucus acini. It's all about salivary gland. Next, artery system, blood vessels. This is a large size artery. Main feature is that we are having more of elastic fiber. These are the thick elastic fibers. These are also called as large size arteries or elastic arteries, as they are having more capacity to retract for blood supply from heart to peripheral tissues. This is the thick band of the elastic tissue is found in elastic or artery or large size artery. Next, coming to medium size artery, we can see the discriminating feature that elastic fibers are almost less. These are the smooth muscle fibers, they are becoming prominent. Compared to large size artery, we can see that this whole band of elastic fibers is more. Next, coming to medium size artery, as I have told, we can find a small band of elastic fibers, but smooth fibers are almost, it's becoming prominent, but elastic fiber is less compared to smooth fibers. Next is vein. Prominent is adventitia. We are having more of the smooth muscles in a vein. This we completed with the artery. Next, coming to the lymphatic system, we are having a lymph node. Main feature is that in lymph nodes we are having germinal epithelium, and these are the main features. The simple things I am saying is that to identify it should be easy, and watching the video should not consume your much of the time. It's about the lymph node. We are having germinal epithelium, where it is much of the staining is found in the surrounding the epithelium. This is about lymph node. Next, coming to thymus. In thymus we will find this trabeculae, and we are having that Hessen's corpuscles in thymus, and red pulp and white pulp is also found. It's about thymus. Next, coming to the spleen. Main feature of the spleen is we should search for eccentric artery. It is eccentric artery means that artery will be peripheral. Means this is the eccentric artery, guys, because this is the eccentric one, the one I marked now, because it is almost edge of this that specific tissue. This is eccentric one. Main feature is that if you mention during the exam eccentric artery is found, means the distinguishing feature of spleen, it will be added to the point for marks. Next is skin, thin skin and thin skin. We are having this thick band surrounding the skin. Mainly in thick skin we are having it's divided into 5 parts. We are having stratum basilis, stratum corneum, stratum spinosum, and stratum lucidium. Mainly one shortcut method is that thick skin and skin can be identified easily. We have to concentrate on this lining, that which is having very thick band compared to the thin skin, which is almost, see, you can see this small line is found in the thin skin, but in thick skin we can find it is almost prominent. And one more thing is that we can't, this space whatever I'm drawn here, we should find this space. Means within 1 minute we have to identify. We can't go for all theory, and there is no time for identifying. Concentrate on this lining and concentrate on this surrounding area. We have to compare. In thin skin we are having many of the things in thin skin. See, so many structures are present in thin skin. We are having mainly glands, rectus pili muscles. So it's about the thick skin and thin skin. In short and sweet, I'm trying to put here. In placenta we are having next is placenta. These are the chorionic villi in placenta. Another one is systematic. Another slide is umbilical cord. We are having it's a easy structure. You can revise yourself, I guess. So the PPT, the video I made is for a short quick revision before going to the exam. It will be very helpful, I guess. If you like the video, please like it, and if this content is good, okay, thank you, good luck for your future.